Hi, our project consisted in proving the importance of worms and other bacteria found in the soil in order to avoid climate change. According to our inquiry, worms and other bacteria, but since we focused especially on worms, are important because they provide the soil with nutrients that make it more fertile and more healthy. And this leads to more plant life, which also leads to the production of clouds since trees produce vapor. And this means a higher albedo coefficient and also less heat. Moreover, soil is a great storage for carbon dioxide since worms and other bacteria feed on carbon compounds found at weeds and leaves near the plants and roots. And these, because what they do is that they absorb these carbon compounds and then they release it, but not into the air, but into the soil. And because this ensures a healthy and diverse population of bacteria near the roots, which protects them. And also, just as a fact, without decomposers, the nitrogen and the carbon cycle would be messed up, and tons of carbon dioxide would be released into the air and water because worldwide 1,200 million tons of fixed carbon is immobilized by decomposers in the soil. And well, without them, all of these greenhouse effect gas would be released into the atmosphere. But worms need to be under certain conditions in order to survive. For example, they need to be moistured but not soaking wet because in that way they would die. And also, they cannot stand really high temperatures over 40 degrees Celsius. And these two factors are related to climate change and they're making the worms population vulnerable since, for example, it's more likely to have water vapor in warmer than cold air, and the presence of water vapor in the atmosphere produces rainfall. So, also, at least here in the city, it's really common to have smog, which is a type of cloud that produces acid rainfall. And now, talking about temperature, it's not that the average temperature will be 40 degrees Celsius, but um, we have to consider that the soil has a lower albedo coefficient, which means that it absorbs more solar energy. So, um, since it's getting warmer, the soil's absorbing more solar energy and worms are dying. So basically what we did for our project is that we collected two samples of soil. One of them contains like about 20 worms, which is this one. And the second one only contains soil without worms. Then what we did is that we poured some vinegar in a bottle and we put some baking soda inside a balloon and we put this balloon in the mouthpiece of the bottle, and when the baking soda fell down, then a chemical reaction took place and carbon dioxide was produced. So we were able to collect this carbon dioxide in the balloon, and with the spring we collected 15 milliliters of carbon dioxide, which we injected directly to each sample of soil. We had to wait like 4 days to take both samples to the chemistry lab, and we did a really interesting experiment which consisted in putting both samples in two different vernier glasses with two vernier sensors in a closed system that detect the presence of carbon dioxide in parts per million, and the results were the following. So what happened with our project is that first of all, we put both samples in these vernier glasses with their sensors. And first, the sample without worms in it was increasing in concentration of carbon dioxide, but like 15 minutes later, the concentration of carbon dioxide was increasing like way too much in the sample of soil with worms in it. And as the professor Adrian Diaz explains us, it's because in a closed system there is a little oxygen and the worms actually breathe. And what happened is that this oxygen was uh, being breathed by the worms, and eventually they started running out of it. So the concentration of carbon dioxide uh, was increasing just because the oxygen concentration was decreasing. Then the soil sample without worms in it started getting a constant concentration of carbon dioxide. So we took another sample of soil fixed by worms and we added it into a third vernier glass with distilled water, and we put the sensor also. And first, it was having a constant carbon dioxide concentration as well. But then we shook the glass in order to break this blocking that avoids carbon dioxide from being released into the atmosphere. And it actually worked because the original concentration of carbon dioxide in this closed system was about 3,853 parts per million of carbon dioxide. And right after we shook the glass, it rose up to 3,900 and a little more, which means that the worms actually absorb carbon dioxide and they fix it, they release it into the soil to make it more fertile and more healthy. And this helps plants near them grow, and also that they lock all of this carbon dioxide away from the atmosphere. And that's all.